Right. So, we've done transcription, we'll now do translation. Okay? So this is the part where we're turning the messenger RNA into a polypeptide chain. Okay? And now I'm going to introduce, and we've talked about this before, the new molecule which is transfer RNA. Right? The clover leaf shaped RNA molecule with a specific amino acid attached. First point, we have now our mRNA strand has left the nucleus through the nuclear pores. Okay? Left the nucleus through the nuclear pores and it attaches to ribosomes. This structure here is a ribosome. Okay? And you know that ribosomes are site of protein synthesis. Right? And this is how it actually does it. The ribosome begins to read the triplet or the codons on the mRNA. And you know that the three bases represent a codon. Okay? As it reads it, the complementary anticodon that is on the bottom of the tRNA molecule lines up next to the codon, the complementary codon on the mRNA strand. Okay? Each anticodon brings with it a specific amino acid. So in this example, UAC codes for a specific amino acid. Let's call it tyrosine, even though it might not be. Okay? So it brings it down to the ribosome and we begin to start to develop our chain of amino acids. Then what happens is the ribosome moves along the mRNA strand, reading the codons as threes. It reads them two at a time. I hope you can see that there's only ever two codons being read by the ribosome at the same time. But as the tRNA brings the relevant complementary anticodon with its specific amino acid, we start to develop a polypeptide chain. Okay? Peptide bonds form between amino acids. As they do, the tRNA molecule leaves to go pick up another amino acid and start the whole process again. Okay? What then happens is this ribosome continues along the strand of mRNA until it reaches a triplet code or a codon called a stop codon. And that stop codon codes for no amino acid and therefore stops the chain. Okay? Now I'll show you that a second way because I have the three other diagrams which are the ones you've got. So hopefully by me showing you these you can work out what is going on. But just like before, the thing about these diagrams is they don't show the ribosome very well, hence why I did that one first. The ribosome's reading the codons on the mRNA. The specific complementary anticodon that is part of the tRNA molecule brings with it a specific amino acid. Peptide bonds form, as you can see. The ribosome shifts along, reading the codon after codon after codon. Once the tRNA has done its job, it leaves to go pick up another amino acid, and we form an amino acid chain that eventually ends when you reach a stop codon. That's what this diagram is trying to represent. This amino acid has no amino acid attached. This tRNA has no amino acid attached, alright? Any questions? Alright, great. Stop. Right, so